Good morning, everyone, and welcome to the second lecture of Jalview. In this lecture, we'll talk about editing, coloring using Jalview. Of course, you can import your sequence, and we will see many methods that enable you to import your sequence for alignment. This will include, of course, drag and drop, paste, or even visiting a URL. With Jalview, you can decorate your sequence with reference and annotation. Of course, you can create an alignment and build shading, conservation, analysis. You can build a tree, principal component analysis capabilities, and many other good features that you will see inside Jalview. In this lecture, we should know how to fetch DNA references for your sequence, how to set feature setting, how to change the view, how to use Jalview to edit your multiple sequence alignment, or even introduce or removing gaps, and of some other useful feature of Jalview. Also, we'll see how to navigate in Jalview using cursor mode or even a normal mode, and different ways to load your sequence, insert your alignment image into your document for publication, Jalview project file, and even you will see how to select, edit, explore, and save regions in your alignment. Of course, we know already that Jalview can work on your computer with a desktop version. Also, we now know that multiple sequence alignment, there is many multiple sequence alignment algorithm in Jalview. So today we'll focus only in the multiple sequence alignment capabilities inside Jalview, because it's actually the program have many other good features for scientists. So why we call Jalview with this name? Simply because it's a Java applet. So Jal means Java, AL means alignment, and view means viewer. So it's a Java alignment viewer. So it's a good for multiple sequence alignment to view and edit and analysis tool. Also, it can run in different platforms, as we already said, in Macintosh, in Linux, in Windows, and other things. It can deal with many numbers of sequences, hundreds of sequences, or even thousands of sequences with minimal degradation performance. And you can load your sequence in many formats, including FASTA format, cluster, MSF, or even PAIR. The next two slides is only about the history of Jalview. You can see that the Jalview have been developed in 1996 in University of Oxford, and then you can take a look at the progress of this development. And of course, all the credits now goes to the University of Dundee for hosting the Jalview website and for developing more capabilities of Jalview. Of course, citing Jalview, if you use Jalview, you have to cite it in your publication. So you can put this two references. So if you will use Jalview in your publication to produce result, of course, for you. When you open Jalview, you will the startup is you will see as example project. In this example project, you will see few proteins that will be aligned for you, and you can see you will see different features in Jalview. So this example will be opened automatically, and then you can see many different features, key features in Jalview. This, of course, including will be see alignment, annotation, structure with JMOL session or trees, or even different things. In the web service in Jalview, this is very important menu, because you can fetch database references. So you have many references, you have many databases, you can fetch your references from these databases. So you can go, if you have sequences and you don't have any references, you can just go to the menu web service and fetch references and choose a database to fetch references from. After fetching the references, you will see something like this. When you put the mouse over any sequence, you can see that a lot of annotations, and a lot of information will appear to you from each sequence. If this is okay with you, it's okay. If this bothering you, you can just from the view window you can click show annotation, or even not or uncheck this annotation. Show annotation or not. If you want to show it, you just keep it's okay. But if not, you can uncheck show annotation. Of course, try to make automatic scrolling. If it's okay with you to use automatic scrolling. If not, you can uncheck it and see with the other way. The good thing in Jalview is in this menu, in the view menu, is a feature setting. Feature setting is very important, and one of the things that I love in Jalview, because you can see many things in the feature setting with different colors. For example, if you have protein sequences, you can color each feature with different color. For example, the signal peptide, you can give it a color different from the chain, from the glycosylation site, from the disulfide bond, splice variant, helix, strand, turn. Each feature of this feature, or each feature of them can be colored with different color, and even you can uncheck any of this feature and choose one of them only to show where exactly in your sequence that feature is. So this will help you to produce your result in the way you want, in the color you want. Even you can set the color or change it whenever you need. Like this, if we just, for example, remove the chain color, so he will show everything, all the features except the chain. And of course, this is very good thing to do in your sequence to show your result. In the format menu, you can usually choose wrap. So instead of seeing your sequence very long and you can just scroll back and forth, you can see it in just one under the other. So if you want to see it in that way, so you can use the order wrap. So you can open format menu and choose wrap. Also in the view menu, also there is two important things to show. If you would like to see the sequencing ID tooltip, you can show the data base reference or not. If it bothers you, you can check or uncheck this. Okay, now we will go to the other part, which is using Jalview to edit your multiple sequence online. Because of course we said that because editing a multiple sequence alignment is so complicated, biologists have to develop text editor that are specific to multiple sequence alignment. If you use any other program like Word document or something to edit your alignment, actually you will have a lot of problems, and this will be very difficult to use. Okay, but that's why this kind of programs have been developed to be able to edit your multiple sequence alignment without having problem. Okay, now if you upload some of your sequences, you will go to and you would like to perform some multiple sequence alignment with it. How to upload your sequence. Uploading your sequence is very simple. So you have many ways actually. You can open the file menu and say input alignment. You can input it from file, if you have a file like a text file with your alignment, or you can import it from URL, or you can import it from text box. Text box means when you press in text box, you can copy and paste your sequence in the text box that will be open when you press text box. Anyway, when you have your sequences already uploaded in the program, you can reorder your sequences. You can keep the order as it is, or you can reorder it. One of the things that help you to reorder is in the calculate menu. You can auto calculate. You can sort your sequence by ID or by length or by group or by pairwise identity or even by MSA ordering. If you write sort by MSA ordering, so your result will appear as in the same order that you uploaded your sequence with. So if you want to keep the same order that you already upload your sequence with. So there's many ways to sort your result of sequences. Sequence reordering is simple. Of course, you can do it by other ways, by highlighting the sequence to be moved. Also, you can reorder with hand instead of using this sort option. You can using by using the your hand. Just you can click on your any sequence and just move the arrows up and down, and you can order your sequence in the order you want. This small exercise is for you to use in your own time, just to see how can you order sequence by using arrows, and see how can we use easily the arrow keys to reorder the sequence. You will see that using the arrows you can simply move sequences from this position to this position only by using the arrow key. Also, when we use after lining and reordering your sequences, actually you can use the web service to use alignment. As we said before, you have many good aligner programs to use. You have TCAFE, ProbCons, Muscle, MAFT, and MSA probes, MGL probes, and others, and Cluster, of course. So you can have it use this program with the default setting, so it will be the same like the website. Also, in this program, it allows you to run TCAFE with preset setting, so it will be much quicker. So this is a preset setting. Also, you have another option with that you can change the setting yourself. If you are an expert and you know what you are doing exactly, so you can change the setting and use the edit setting and run. So you can change the setting of this program. All this can be found in the web service alignment, and then change with the setting as we can see now. But when you change the setting, you will have something like this, and of course you use your experience and this and choose what you exactly need. Of course, this is beyond this course, so but this is an ability in Jalview that you not only using the default setting, but also you can change the setting what you want. And the coloring scheme, actually, as we will see later, that the cluster, the Jalview have many coloring scheme, including Cluster X, and this is one of the good coloring scheme that I prefer to use, but and understandable for everybody. But if you want to use other coloring scheme, there is many other coloring scheme, as we will see later in the lecture. One of the good things in Jalview when you're presenting your multiple sequence alignment is hiding region or presenting some region and hiding others, so you can actually emphasize in what you would like to show to the editor. To other people, for example, if you would like to show other people only the conserved part, only the active domain, only the active site, so in this regard, so you will hide other region and keep your special segment of your sequence. So you need to use the options for hiding regions. Hiding region, as we said, excludes some of the sequences without actually deleting them. So you can exclude some sequence, but you will not delete it. Of course, this facilities is good to be used to create several different views, of course, in the example alignment file. We will see how to do this, and how to hide region, and how to not hide region. For example, if you would like to hide a set of sequences, you can simply click. You can select them first, and then select. You can right click and choose hide sequences. So by this way, you will hide sequences. And then like this, you can see that you select some sequences, number 1, number 5, number 6, whatever. You can choose whatever sequence you want, and then choose the option hide sequence. If you take this, of course these sequences will be hidden. But if you press in the blue triangle here, if you press in the blue triangle and press on the reveal all, okay, this hidden sequences will come up again. This is the way to hide sequences. Also, you can hide column. You can also select columns and then right click and then choose hide columns. And the same thing, if you see a blue triangle, you also, if you check this blue triangle, you can bring it back again like this. So you can choose a few columns, and then you can right click and say and press on hide columns. If you would like to reveal, you will see a blue triangle here. If you just right click on this blue triangle and press on reveal, this few columns will appear again. So now this is the two methods to hide columns and hide sequence. Okay, you can also view only a small part and hide other things, including columns and include sequences. So if you would like to select a special region, in this special region you would like only to show that, so and hide any other sequences and columns other than this selected region. So you can simply do what? You can simply click on the region that you would like to show only, and then right click on the view. From the view menu, choose hide, and then from the hide option you can choose all but selected region. And it's simply, you can do this by Shift Control and H. Shift Control and the letter H. If you press this, it will hide everything except the selected region. This is a very good feature in this program. We use it many times just to show a specific part of your sequence that include sequence and include columns, only very specific region of your sequence. And of course, if you would like to reveal it again, as we said before, in the blue triangle, you can right click on the blue triangle and then press reveal. This is the way to do that. It's only very easy. You can choose the part you would like to show, and then just right click, open the view menu and select from the hide option. You can see all but selected region. Of course, by hiding some region, this will help you to easily see a specific part and show some other and hide other parts that is not needed. So you can even actually, by doing this, you can have parts in your alignment that would like to be aligned properly. By doing this, you can align some parts and exclude other parts in your sequences. When you do this and do the alignment, you will have your result like this as a segment. If you choose parts, so actually this will make your alignment as different segment like this. As you see, that every segment of your sequence, every few column of your sequence have been aligned independently. So each few column, when we do the hide region within your sequence, as the arrow shows, so actually the alignment happen independently within these sequences. As you see, this part have been aligned independently from this one, from this one, from this one. So it's like sectioning your alignment. It's like sectioning your alignment and your sequence, so you can see each part aligned independently from the other one. Why we need to do that? Of course, this is if you are satisfied with the alignment of part of the sequences. Hiding it will preserve it, while other regions are optimized. This is one of the good reasons for doing it. The other reason is if you are interested only in a small part of the alignment, hiding the rest will allow you to align only the local visible region. Also, the third reason for doing hiding alignment is hiding regions acts as a boundaries, imposing constraints in the multiple sequence alignment. These are three good reasons for doing this hiding tip. Representing a group with a single sequence. Actually, this is a good thing to do if you would like to put a name of a group of sequence and put an ID, and you would like to this sequences to represent something. So it's very simple. You can see this by the coming example. You can take the selection, what you would like to select, any group of sequences, and then right click and take and choose selection, and from selection option you can take edit group, and then you can choose edit name and description of the current group. So you can say, as we said, choose a group of sequences and put a name for this group of sequence. You can name it whatever name you want, so you can identify this group of sequences from the others, so you can remember what this means. You can also introduce or remove gaps simply by using entering by pressing by the keyboard. So you can, of course, this very easy to do. You can just press dragging residues with the mouse when shift or control, and you can add gaps or even remove gaps from your alignment. Of course, if you move to any part, you can cut or even add with the keyboard. You can add more sequences, you can add more letters, you can delete. It will move the sequence that you edit will be only changed, but not the others. So that's why it's a very easy to do that with the multiple sequence alignment. You can easily choose a small part, for example, and say edit, choose edit from the menu, and then cut or copy or edit or whatever you can do with your sequence. This is a very simple way to remove or add gaps to your sequence. Of course, locked editing, this is another thing you can do. That this is a very good option if you don't want to change the alignment, so and do different things without changing alignment. This is called locked editing. This is very useful. You can of course reach this from the edit menu. Some other features, useful feature, of course, this will include calculate consensus. This you can find in the calculate menu. You can auto calculate consensus. This will allow you to see the consensus of your sequences, and this is a very good feature also to see which part is preserved, which part is conserved, and which part is not conserved. Removing redundancy, this makes sure that no pair of sequences is more than X% similar. So if you would like to remove redundancy, this option can be found in the edit menu. From the edit menu, you can go to remove redundancy option, and this will allow you to remove any redundancy from your sequence. As we said, of course, calculate tree, and there is many different algorithms to make a phylogenetic tree. This, of course, differences between all of them will be you will understand the differences between them, of course, in the next module. But in this module, we only show you that Jalview can produce a tree for you and can calculate the tree for you using your multiple sequence alignment. This is practical. In your practical, you can try to open the BFAM accession number and try to see and use it in the program, and try to follow these steps